Hello everyone, and welcome to my lecture series on bioinformatics. And let us continue with our demonstration on molecular phylogeny. So in our previous lecture, I have shown you as to how do we download software for multiple sequence alignment. I have shown you from where you should download your ClusterLex program and how do we use it. Right, so let us now continue the same discussion and try to build up a phylogenetic tree for the multiple sequence alignment that we have already generated in our previous lecture video. Okay, so this is your multiple sequence alignment. We are going to revisit it in a while. So first thing first, I will first show you as to from where do you obtain the PHYLIP software. So to obtain the PHYLIP software, just search for PHYLIP in your Google. Okay, so you would be taken this is the homepage for PHYLIP at University of Washington. This is where you need to go. Okay, and over here you have got link get me PHYLIP. Okay, you would be carried to this page, and then you are supposed to, there are two options over here, and as per these two options, if you have 62-bit operating system, which are very common nowadays, then you are supposed to download this. My system is a 64-bit system, therefore I have already downloaded, therefore the color of this link has changed. If your system is 32-bit system, then you are supposed to download this. But there is a catch over here. If you download 32-bit binaries or 32-bit version of this PHYLIP software and try it to run on 62-bit, you on 64-bit operating system, then you might find it difficult. Okay, some programs might not work. So it is better that you should know what type of operating system do you have. So if your computer or laptop is new, most probably you are working on 64-bit Windows system. So just ask someone who is familiar with computer, and then download an appropriate version of PHYLIP for your own system. Okay, so all you need to do is to right click and save link as. Okay, or you, if you simply click it, it will be downloaded as a zip file. Okay, so most probably it will get downloaded in your downloads folder. So I have already done it, and you would get a zip file which would look something like this, PHYLIP 3.6.98. so this is the latest version that we have. Okay, and what you are supposed to do is to, then you are supposed to extract your PHYLIP into say some folder. Okay, you can have any of the extracting software installed. We'll say for example over here I have installed this WinRAR. 7-zip is one of the open source freely available software. You can download that 7-zip that I have also installed over here, and you can use this 7-zip for any type of uh, packing, unpacking, or archival stuff, right? So either way, you can either use 7-zip or WinRAR or WinZip, whatever software you have. Extract the content of this zip file in say some folder. Okay, so if I use this option, extract to PHYLIP 3.6.98, that means this software will create a folder named PHYLIP-3.6.98 and extract all the files that are present in this archive file. So this is how the output would be. Okay, so once you have extracted everything in a folder, you can open it up. There is another folder called as PHYLIP, and you have got three subfolders and a web page over here. This web page is you know uh, documentation file. Okay, so it gives you an idea about what these programs are. So this is main documentation file. So it says what are the different programs that are present in this package. Okay, drawgram, draw tree, these are all tree drawing, consensus, tree editing and tree distances based software like drawgram, draw tree, consensus, tree dist, retree. Okay, and you have got some molecular sequence methods like protparse, which perform parsimony. Then you parsimony on protein sequences, DNA parse, which perform parsimony on DNA sequences. Okay, so on and so forth, right? So this is documentation. Of course, you can click over here and you get all of the information on how to use this software and what are the expected input and output, on what basis this software has been designed, right? So this is the documentation stuff. Okay, uh, this is this is for documentation. Over here src stand for the source folders. Okay, and this is what we are interested in. This folder called as exe contains the executive files for all the software that are distributed in this package. Okay, okay, so here we have at least 48 atoms. Okay, but these 48 atoms contains some different files, like there are some jar files over here, executable jar files, and there are some font files, as you can see over here. So we would require these files. So this draw tree, drawgram, these jar files were are new in this 3.698 version. Okay, these are Java based implementation of programs like drawgram and draw tree. Okay, so if you want to use these version of drawgram, then it is necessary that you have already installed Java, right? If you want to use this Java based version, right? And if the proper version of Java is not installed, then you might fall into problem. It might not work very well. So we would be using this simple drawgram program. Okay, so now this is about your PHYLIP package. Where do you obtain it, and where is the documentation for your help purpose, and where are the computer program that we would use in this presentation or in this demonstration. Okay, we would use one by one most of the programs from this package. Okay, so now I hope you are familiar with this PHYLIP package. Okay, now let us start thinking step by step that we have learned in our theory lecture. Okay, so let us begin with again discussing. There is no harm in again listening to the stuff where we have to decide what kind of method we would be using for creating a phylogenetic tree. Well, I'm doing this because some of you might directly end up at this video. Okay, you are advised to see my previous video and understand those things in more details, and then use this video lecture for constructing the phylogeny. Well, in brief, first we need to decide, once we have this multiple sequence alignment, based on the data from this multiple sequence alignment, we have to take a decision as to what kind of tree reconstruction method we are supposed to use. Okay, so how do we do this? How do we interpret this data and then decide what kind of method we have to use? Okay, so it is very simple. So this star corresponds to the conservation in your multiple sequence alignment. Now, if you see that there are over here 590 columns in your multiple sequence alignment. Okay, so if you find out that out of these 590 columns, if there is a situation like 95% of time or 90 more than 90% of time, if more than 90% of your columns are highly conserved with the star over here. Okay, if you find star above more than 95% of column, that would be an ideal case to use maximum parsimony method. Okay, why? Because maximum parsimony method requires very requires variation to be very small. Okay, the situation of or in other words we can say that it requires high level of conservation. Having high level of conservation is another way of saying having small level of variation. Okay, if the variation is small, or alternatively if the conservation is very high, that would be the case you would be using maximum parsimony. So of course now you can see this data and you don't find high level of conservation, only very few of the columns are having this highest conservation. So I don't think this is an ideal data set to be used for maximum parsimony. Another reason to reject maximum parsimony is the number of sequences. Okay, what I have told you in our previous lecture, that if there are around 12 or we'll say for example 15 or even 20 can also work sometimes. So if that is the range of number of sequence that are present in your data set, then you are supposed to use parsimony. In our case, clearly we have more than 30 sequences over here. So these are the two reasons that we are not using maximum parsimony. So we have completely discarded this. So we are not using maximum parsimony. Therefore, there are two main type of tree reconstruction methods left. What we call it as distance-based method or maximum likelihood method. So when do we use distance-based method? We use distance-based method when the variation among the sequences is present. That is, some sequences are more alike than others. Now check it out. As you can see over here, that most of the sequences are similar, while some of the sequences are dissimilar. Okay, so this is an ideal case. Some of the, I mean, when the amount of variation is intermediate. Okay, and this distance-based method are very likely to be used when you deal with large number of sequences. Okay, so we have got 33 number of sequences, and I think this is the ideal methodology that we should implement for performing phylogenetic analysis. Okay, so now we also cannot use the third said method, that is maximum likelihood. Maximum likelihood is to be used when maximum likelihood method is to be used when uh, when there is you can say sequences are more variable. Okay, when your sequence has got enormous level of variability, that means a very sparse stars are present in your multiple sequence alignment. Okay, that would indicate that there is high amount of variation in your sequence. Okay, but we can clearly see that uh, amount of variation is intermediate in this case. Therefore, it won't be logical to use computationally extensive method like maximum likelihood. That would require a lot of computational time and memory to uh, for computing the phylogenetic tree. And moreover, as it says that that computational complexity increases with the number of sequences. We already have 33 sequences. Therefore, as the number of sequences increases, well, that increases the computational time and computational power that is needed to be used. Okay, so of now analyzing this multiple sequence alignment data, we can clearly say that it would be probable that we should use this distance method, and this type of test is needed to be required when you perform such phylogenetic analysis. At the step of multiple sequence alignment itself, you should decide what type of method do you want to use. Okay, and we have discussed these things in quite detail now. Okay, so now let us start discussing a few things more, right? So we need to perform reliability test. Okay, therefore we have to perform this bootstrap analysis. Okay, so that we would be in a position to say ki whatever results that we are presenting are reliable. So to test the reliability, we will have to perform bootstrap analysis. Okay, and we are supposed to generate a rooted tree. Therefore, if we want to generate a rooted tree, we would also have to take care of an outgroup. So how do we do this is going to be uh, you know uh, an interesting thing to learn. Okay, so what is that we need to do? We need to perform bootstrap analysis for reliability. Okay, we need to decide a proper outgroup for routing the phylogenetic tree. Okay, and third thing that we need to do is that what we were discussing, what method to use for phylogeny, right? And we came to conclusion that could be using distance based methods. Okay, so this is what we need to do. So we need to perform bootstrap analysis and test the reliability of the tree, and we need to decide a proper outgroup since we would be needing a rooted tree. Okay, so first let us see how do we handle this situation of deciding the outgroup. Okay, so in this case, again I would like to go back to your data set. Okay, here is your data set, and this is the file where I have placed all my sequences. Okay, moreover, this is the details of the codes and the complete name of the NS5B protein. Okay, so in our data set that we are going to use, there are, say, some how many some 31 sequences from hepatitis C virus. Okay, and I have included the NS5B protein from hepatitis GB virus as so that we can use this NS5B protein from hepatitis GB virus as an outgroup. And that is why, my friends, I kept the code HC for hepatitis C virus and HG for hepatitis GB viral agent. Okay, so now what is the logic behind selecting this hepatitis GB? So, a candidate from hepatitis GB virus was selected as an outgroup in this analysis because the GB agent is known to be distinct from currently known hepatitis virus, yet it is related to them. And hence, this fact make it a suitable candidate as an outgroup. Okay, so in order to guarantee the consistency in rebuilding evolutionary events, NS5B protein from hepatitis GB virus, the closest living relative to human hepatitis C virus, was included in this analysis as an outgroup. Okay, so now you should learn a fact that uh, while constructing your data set itself, you would have to include an outgroup in it. Okay, so let me show you. These are all, initially I have placed all hepatitis C virus sequences, and at the end of this list I have placed this NS5B protein from hepatitis GB virus. Okay, so this is how you intentionally include an outgroup in your remaining in-group. So your objective is to find out how the sequences in the in-group have evolved. In order to do that, you would need some outgroup to root the sequences or to root the sequences in your in-group. So you would have to include the outgroup right at the multiple sequence alignment uh, step itself. Okay, so now you know that what will be the outgroup for our hepatitis C viral sequences and why we have included that particular outgroup. Okay, the ideal property of an outgroup is that it should be related, but it should be distinct from the in-group sequences. Okay, and it should be close relative to the given set of sequences. These are the ideal conditions for an outgroup to be selected. Okay, so this is how we deal with the idea of we need to decide the proper outgroup. Okay, so the outgroup in our case would be the NS5B protein from hepatitis GB agent. Okay, so let's so we have taken care of these two things. Now what method to be used? We have decided now that we would be using distance based method. I would remove it, and we need to decide proper outgroup. We have already included that outgroup in your multiple sequence alignment, and we would be using it appropriately as and when it is required. Okay, and we also need to take care of reliability, and for that we would be performing bootstrap analysis. And how do we do that? I would just demonstrate to you in a while now. Okay, let us begin our phylogenetic analysis and see how things will work. So now let us come back to this slide. Okay, so how do we do this bootstrap analysis? We do this bootstrap analysis by reshuffling the columns in your multiple sequence alignment. Okay, so whatever columns are there in this multiple sequence alignment, what are we going to do? We are going to reshuffle it and create something called as the pseudo data sets. Okay, over here, for the sake of timely completion of this tutorial within 1 or 2 hours, instead of generating 1000 pseudo data set, we would generate only 100 data set. Okay, so when you yourself perform this tutorial at your home, you can generate 1000 data set. And I also need to take care that this practical finishes in some finite time. So for the sake of simplicity, instead of creating 1000, we would deal with only 100 replicate. Okay, so what we need to do is to create now 100 replicates. Ideally, people rely on the data set which contains some 1000 pseudo data sets. Okay, so now how do we do this, right? So as I have already mentioned over here, there is program called a sequence boot that would generate some 1000 multiple sequence alignment. In our case, we would be generating 100 multiple sequence alignment. Okay, so let us peep back into our PHYLIP package in our executive file. Okay, we have this program called as sequence boot. We would need this sequence boot program. Okay, so let us copy it. Okay, and create a new folder. This is the first step, therefore I will give it a name as 1 and sequence boot. Okay, so over here we would paste our sequence boot program. So this sequence boot program will perform the function of reshuffling of the columns in your original data set and generate 100 pseudo data sets. Okay, for this we would require our input sequence, right? So let us go back in multiple sequence alignment, and this is the file that is to be used as an input for your for your phylogenetic analysis. If you remember, these are the files that were generated when we run our clustalex program. Okay, so let us copy this program, copy this data. Okay, so remember, the file which has got this .phy extension is the file that is required as an input for PHYLIP packages. So we have copied it, and this is our first step. We will paste it over here. Okay, one more thing that I need to explain is that why we have given those codes. Okay, so this is the multiple sequence alignment in your PHYLIP format. Okay, so now the thing is that your PHYLIP allows only 10 characters in your comment line. So this would indicate if the name of your sequence contains more than 10 characters, it would be trimmed off after the 10th character, and only first 10 characters will be included in the file name. Therefore, if you use whatever default lines that are present in your FASTA format, well, those are of complicated type. Okay, and at the end of your phylogenetic analysis, then you would get confused as to which one is what sequence. Okay, therefore, to avoid such disaster at the end of your phylogenetic analysis, it is advisable that you give some codes which are less than 10 characters. Okay, so you might now start to note that this each and every code that I have given, it contains at max 10 characters. Okay, so this is the reason why we have to assign some codes that you would understand, right? Why? Because your PHYLIP format itself allows only 10 characters as to be given as a name for your sequence. So that is why we have assigned or devised these codes that we have used in our phylogenetic analysis. Okay, so this was the reason for deciding those codes. And make sure that in your code there are only 10 characters. Why? Because if there are more than 10 characters, your PHYLIP program is going to eventually trim off whatever is present after first 10 characters. Okay, so that is why we have to have sequences with at least with at max 10 characters of uh, 10 characters in its comment line. Okay, so make sure you do that so that you can keep track of which one is what sequence. Okay, so that was the reason for giving up that codes. Okay, so now let us perform this bootstrap analysis. What we would be doing in this bootstrap analysis, we would be generating some 100 data set by reshuffling the columns in your multiple sequence alignment. Okay, and to do this job, uh, the program that would be needed from this PHYLIP package is sequence boot. We have copied this program and we have pasted this into a separate folder. Okay, now let us double click it. Okay, it might appear that there is some error, but that is not the case, my friends, because if you remember in your theoretical sessions, I have told you that by default PHYLIP program would look for in file or in tree as an input file. So this line indicate that it is unable to locate an input file with name in file. Okay, and that is true. Why? Because the name of our input file is sequence with outgroup.phy. It is not in form of in file. Therefore, we have to tell over here as to what is the name of your file that contains your multiple sequence alignment. So we have to give this name over here. So it is asking for what is the name of the file that contains multiple sequence alignment. Okay, so sequence with outgroup.phy is the file that you should look. Okay, so still we have done, not written, it should be sequences sequences with outgroup.phy. Yes, now things are working. It has detected, okay, this is the file which is needed to be uh, used. Okay, so how do this program works is quite very simple. This is the command line version of the sequence boot program. Okay, so what is given over here is say some codes. Okay, so well, say for example, what we are supposed to do, we are supposed to create 100 replicates. Okay, and now when you would work on a live case, and instead of 100, if you want to generate say some 1000 replicates as it has been mentioned over here, instead of 100, if you want to generate 1000 replicate, then you would have to change this value of 100 to 1000, right? Then and then it would generate those 1000 replicates. So how do you change it? If you want to change this setting of how many replicates, you would have to find out the corresponding code. The corresponding code for changing the number of replicates is R, as you can see over here. Therefore, all you need to do is to type R and hit enter. Now it would ask you as to how many replicates do you want to generate. So we would give the number 1000 and hit enter. Now you can see the value of how many replicates has changed from 100 to 1000. Okay, but for time being, since we are working on only 100 replicates, we would revert back to its default of 100 replicates. So this is how you need to do. Okay, so if you want to change any setting, you need to identify what code we have to insert. Okay, and accordingly it would ask you as to what is needed to be generated. Okay, so this would indicate that we would be generating 100 replicates. We are working on IBM PC, that is also need. You would we are generating entire data sets, not just data weights. Okay, so if you want to, well, say for example, write only weights instead of data sets, you would change the option S. Okay, now it would write out just weights. Okay, but we need to generate data sets in entirety, right? Therefore we would again revert back to S. Now we would be generating entire data sets. Okay, so now I am happy with these settings. Okay, we have inputs of molecular sequences. Okay, we are performing bootstrap analysis, right? We would be using regular sampling fraction. Okay, so on and so forth. So if you want to go in more details of what these are, maybe you should look out in your documentation section. Okay, PHYLIP's documentation section that I have already shown you, and check out for what is there in sequence boot. Okay, so you get an idea about what is bootstrap, what is partial bootstrap, right? What is jack niffing, so on and so forth. Okay, so this is where to look if you don't know what exactly these settings mean, right? So I'm happy with this setting now. Therefore it is asking you, hit Y to accept these changes, or type any letter as provided in this column to change the setting. Now we are happy with these settings, so I would hit Y and hit an enter. After pushing enter, it would ask you some random number seed, and it must be an odd value. So I would give it some odd value, 1 2 3, and hit enter. Yes, this is it. Okay, it has generated all of these 100 replicates, and all of these 100 replicates are saved in the file name as out file. Now it is asking press enter to quit. If I hit enter now, this computer program would shut down. Now it is gone. So now let us check in our sequence boot. It has created an out file. Okay, so now this out file, you can open it up in TextPad, and you can see now there is 33. This is the you can say that this is the comment line for your first data set. Okay, 33. This is, I will just show you that we would search for 33 next. Okay, this is first data set. If I find next 33, okay, so over here your next data set has begun. Okay, this was the first data set, this is your second data set, and of course if you scroll down, this is your third data set, this is your fourth data set, right? And you can see that the columns have somehow changed. SSS is the first three column over here, and by the end you can see the data sets are different, right? The column over here is MMST, right? So in this way, SWW, we have got some different column. So in this way, what we have done over here is that we have kind of rearranged these columns and generated those 100 replicates as shown over here. And from each replicate, each 100 replicate that we have, we would generate a phylogenetic tree from each replicate. Okay, so that would be our next task. From each pseudo replicate, it's time for us now to generate a phylogenetic tree, and this is what we have. We have these 100 replicates in the pseudo data set that we have just generated. Okay, so this is the out file. Okay, now what to do next? Let us follow our same protocol. Now we have come to this step. From original data set of multiple sequence alignment, we have generated 100 multiple sequence alignment. Okay, so now what is the next step? We would be using this prot-dist program, which would generate 100 distance matrices from 100 data set. Okay, so we would be using this prot-dist program now. Okay, we need to generate 100 distance matrices now. Okay, so the second step that we would need to do is to create a new folder, name 2. We are using this prot-dist program, right? So in this prot-dist program, the output of sequence boot will be input for your next subsequent program. I would copy this out file and place it here. Okay, now we need to copy that prot-dist program from our executives, from our PHYLIP package. You have this prot-dist program which is located over here. I would copy it and I would paste it in your prot-dist folder, right? And by default, your PHYLIP program would look out if there is an in file that is present in the same folder, which would contain the input. Therefore, I have renamed this out file to in file, so that when we double click on this prot-dist program, it would directly take this in file, and by default it would create out file as an output. Therefore, we need to rename the input for this computer program. Okay, so now it's time that we double click it. It would give the settings menu, which is shown over here. And now I will tell you how this distance matrices differ from the distance matrices that are generated in cluster. Okay, so first and foremost, the evolutionary models are not implemented when you generate the distance matrices in cluster, and therefore the tree that is produced from neighbor joining algorithm that we have used in our cluster do not account for any evolutionary inferences as such. But on the other hand, when we generate distance matrices for the multiple sequence alignment, we are taking account of some evolutionary model. Okay, so you can use different evolutionary model for creating this distance matrices. You can use this Jones-Taylor-Thornton matrix, or you can use this PAM, or you can use this Kimura, or you can use this PMB. What are these? You can check it out in the documentation file for this root dist. Okay, so for our sake of simplicity, we would be using the default program or default evolutionary model for creating a distance matrix. Okay, so now we need to change this thing, M, analyze multiple data set. Over here by default it is no, right? But we have 100 data set, therefore we need to tell that there are 100 data sets that are needed to be used. Therefore we need to change this M option. Okay, now it would ask you whether you have multiple data sets or you have multiple data weights. If you have multiple data sets, then type D, or if you have multiple data weights, you are supposed to type W. So what do we have? We have data sets. Since data sets is something that we have generated in our previous step. Therefore, I will hit D, hit and enter. Now it is asking as to how many data sets are there. We have 100 data sets, 100 pseudo replicates. Okay, now yes, we have 100 data set, and we are analyzing multiple data sets. Okay, and I think this is what we need to change here. Okay, so I will now the settings are correct. Yes, let me see first. Yes, yes, the settings are correct. I am satisfied with it. And now we are creating distance matrices. Now this shows you this kind of this program shows you at what data set we are currently at third data set. That means whatever whatever 100 data set that we have generated, for each data set it is now calculating the distance matrix, and it would take some 10 minutes to generate all 100 data set. So this is where we are. From these 1000 multiple sequence alignment, we are now generating 1000 sorry, from the 100 data sets we would be generating 100 distance matrices. Okay, and after this step is complete, we would be using this neighbor program for calculating 100 phylogenetic trees from these 100 distance matrices that are being generated now. Okay, so you need to be patient for a while. Okay, and we would be considering the data after 100. So we are at 13th data set now, and it is computing the distance. Okay, mind that we are using JTT evolutionary model for creating these distance matrices. So if you are still here, you can skip to after 10 minutes. Let it compute. Okay, we are still at 50. It will take 5 more minutes. So we have reached at dataset 77. Just we need to be more patient for a while till we compute all of these 100 distance matrices. So we are on 96th data set now. After 4 data set, our data for distance matrices would be ready. And next what we need to do is, from this 100 distance matrices that we are generating now, I would tell you how do we use this neighbor program to generate 100 phylogenetic tree. Yes, we are done now. Now it is instructing us that the output has been returned to the file name out file. It is done calculating and we can press enter to quit this program now. I am pushing this enter button, and once I push this enter button from my keyboard, the computer program would stop. Okay, now let us see how this out file of your prot dist looks like. Okay, see, this is the distance matrix for first data set. Okay, as you can see over here, all of these are distances with respect to other protein sequences. This is first distance matrix. Okay, similarly we have second distance matrix that start from over here. This is the distance matrix for second data set, second pseudo data set, and there we have from here the distance matrix for the third data set starts. Okay, and in this way this is a big file which contains 1000 distance matrices, right? So this is the 100th distance matrix. Okay, so this is how the data is arranged in the out file that you obtained from the prot dist program. Okay, so now as I have already mentioned, we are supposed to use this neighbor program as per our protocol. We would be using neighbor program for generating 100 phylogenies from freshly generated 100 distance matrices. Okay, so besides this neighbor program, there are other distance based methods available. So if you wish, you can also use distance based method like programs like fish and cache. But in our paper that I have described, we have used neighbor as a program for generating 100 phylogenies. Okay, so in this demonstration we would be using the same program. Okay, now it's time for us to look out for this neighbor program in the executives. Look over here, we have, you can use this neighbor program, or you can use this cache program, or you can use this fish program for generating the distance for generating the phylogenies. So here it is. We need to copy it, and since we are working on this third step, that is generating phylogeny, we would paste it over here. We would require the input in the same file, and we are going to take input from the output of our previous step. Okay, so what was our previous step? Our previous step was prot dist. We would take its out file. This is the output of the prot dist. We would copy it. We would paste it in our neighbor program. Since after we run this program, it would look for in file. Therefore, we will rename it from out file to in file. Okay. When we double click it, it would start. Okay, so now this neighbor program implements either neighbor joining algorithm or UPGMA as a clustering algorithm to generate the phylogenes or phylogenetic tree. Okay, so you can select whether you can use neighbor joining or UPGMA from by selecting N. Okay, if you hit N, you can toggle between this neighbor joining or UPGMA. Okay, for our purpose, we would be using neighbor joining itself. So let us revert it back to the default neighbor joining program. Okay, so here we go. Now this is very important. It is asking what is the number of outgroup in your data set. This is really very important. By default, it will use whatever sequence that is present at position 1, it would use as an outgroup. Okay, but for your information, in our data set, our outgroup is located at 33rd position. Okay, I would show you in your original data set sequence with outgroup.phy. This hepatitis GB NS5B protein is always placed at the 32nd position. Okay, sorry, 33rd position, right? This is your original data set, I suppose. In original data set, your outgroup is placed at 33rd position in your original data set, as well as in your whatever you have obtained from sequence boot as well. The out file that you have obtained from sequence boot, this outgroup is placed at 33rd position. This is your first data set. In your second data set as well, here start your second data set. In your second data set as well, it is placed at 33rd position, and in subsequent data sets as well, it is placed at 33rd position. Well, now let us check out in our last data set whether it is placed at the same position or not. Okay, look, in our last data set it is placed at 33rd position. Therefore, we need to change this setting. We need to tell that our outgroup is placed at 33rd position in our data set. So to change this setting, we would have to type its corresponding code. The corresponding code for changing the outgroup is O. Type O, hit enter, and now we need to tell over here that our outgroup is placed at 33rd position in all of our data sets. Okay, now it has changed. Yes, we are using outgroup and it is the species at number 33. Okay, so we would be, yes, we also need to change this option M that corresponds to multiple data set. By default it has got no, but we have got 100 data sets with us, right? Therefore we need to change the option for M. How many data set do we have? We have 100 data set. Hit and enter. It is asking now a random number 1 2 3. It should be odd. Yes, this is what we need to do, right? And we don't need to change anything else. Yes, I am satisfied with this. I would type yes to accept these changes and hit enter. Yes, now we have implemented this neighbor joining algorithm and it has created two out files. First is out file, another one is out tree. Okay, it says that it is done calculating, and after hitting enter we can quit the program. Now I am hitting this enter button on my keyboard so that the program will end. Okay, now let us check in neighbor. Yes, it has written this out file and out tree. So this out tree, let us visualize in textpad. This out tree would now contain 1000 phylogenies. Okay, this out tree would contain 1000 phylogenies. Let us see how they are represented. So this is your phylogenetic tree for the first data set, okay, where outgroup is located outside, right? Then this is your second data set, the phylogenetic tree for second data set. Over here outgroup is located at the outside. This is your third data set. Over here outgroup is located on the outside, and subsequently you have this 1000 phylogenetic trees, and in this phylogenetic 1000 phylogenetic tree, every time the outgroup is located on the outside. That means we have successfully used HGBB as an outgroup for routing your phylogenetic tree. So this is where we are in our protocol. From 100,000 matrices, we have now generated 100 phylogenetic trees. Okay, so this is how they look like, and this out tree is your 100 phylogenetic tree in NIVIC format. Okay, so this is the output that we are going to subsequently use. Okay, so from this 100 phylogenies that we have generated, we would now generate a consensus tree, and a single phylogenetic tree would be created with weights or reliability weights on the internal nodes. Okay, so for that purpose we would be using consensus program. Okay, so this is the out tree that would be used as an input for your consensus program. Okay, so these are the output from your neighbor. So let us copy this out tree. The fourth program that we are supposed to use in our protocol is consensus. Okay, we are going to generate a consensus tree from the 100 output trees that we have generated. Okay, and the input is required in NIVIC format. So let us find where that consensus program is. Over here we can copy this consensus program, okay, and paste it over here. When we run this consensus program, by default it would look for and it would look for an input which is called as entry, and now it's time for us to double click it. Here we have got its setting. Okay, so let me tell you what options is that we need to change. Okay, so this C option let you decide what type of consensus tree you want to create. If you want to create a strict consensus tree, that is what was advised uh, in this particular figure. You are supposed to create a strict consensus tree. From this is what we have done. We have used sequence boot to generate 1000 triplicate. We have used neighbor program to generate these 100 phylogenies, and we would be using consensus program to create a consensus tree. In this particular example, they have mentioned that a strict consensus tree is needed to be created, but there are other options also available. You can create this MR consensus tree, or what we call it as majority rule consensus tree extended. Okay, and you can check out in its uh, documentation as to what are these trees. Okay, so we would be using the default majority rule extended as a consensus type. Okay, so over here we would also have to tell that we are supposed to use this outgroup, and the outgroup is present at position 33. Okay, that is all. We don't want to change any other settings. Okay, so are these settings correct? Yes, they are correct. Okay, that is it. It has generated a consensus tree, and it has written the consensus tree in a file name out tree, and in out file it contains the data in NIVIC format. Okay, so it says that it has done a calculation, and we can press enter to quit this program. Okay, now let us see this consensus tree that we have generated, how it looks like, and where are the reliability weights. Yes. Okay, so let us open this out file in textpad. Okay, now reliability weights are provided at each and every internal node. So what do this indicate? This indicate that out of 100 trees, 97 times this HC1AH is found to be sister with HC1A1, and 100 times this HC1BJ1 was found to be placed near to this HC1AH and HC1A1. Okay, so now these number indicate that the reliability of this much portion of my tree is very high. Look, you can say that there is reliability weight of 197, right, as compared to this value of 21. So out of 100 times, it was only 21 times it was found that these two sequences are placed to each other. Okay, on the other hand, the reliability of having this tree is very high, this much portion is very high. So in this way, you can see that based on this reliability weights, you can decide what portion of my tree is more reliable than other portion of my tree. This is how it is supposed to be inferred. Okay, now in in many papers you won't find such a tree that is shown in papers में ऐसा tree नहीं रहता है, right? So in my paper itself, let us see how we have shown a tree. So now our objective is to generate a tree that looks something like this. Okay, so we have tree which looks like this, but for publication you would want to have some tree which looks like this. Okay, more good looking tree. And in order to generate a tree like this, we would be using program called as draw tree. So we would be using the consensus tree which contains a single phylogeny. We would give it as an input to program called as drawgram and generate a final rooted tree. Okay, let's do this. So this would be our final step in phylogenetic analysis, to generate a consensus tree. Okay, so new, you generate a last folder 5 drawgram. Okay, let's look out for the executive for drawgram. This is what we have. Would copy it. Okay, and paste it over here. For this we would require the output in NIVIC format. Yes, this is what we want. and we would also require a font file, something. You can use any font file from over here. Okay, let's use this font file. Okay, so we have everything in place now. To run this drawgram program, it would need this font file, and when we run this program, by default it will look for in tree file. Okay, let us now run this program. So it says that it cannot find the font file. We need to provide the name of the font file. The name of the font file is font font1. Okay, here we have. So we need to do a lot of changes over here. First, we need to decide what type of screen are we printing. We are working on IBM PC. We don't need to change this. Okay, final plotting device is PostScript printer, which is friendly with Unix based system, but we have Windows based system, therefore we need to change this final plotting device. So to change this, since we are working on Windows, I have to hit that P button, and let us see over here what is compatible with Windows operating system. Yes, we have got this Windows bitmap. We can use this W option and select a file compatible in Windows bitmap. Okay, now it is asking how much your tree should be on x-axis. Let's say, for example, let the tree to be 100 points on x-axis and 2000 points on y-axis. Okay, then increase the tree, let the tree grow horizontally. We have got different tree style. Okay, by default it is phenogram. You can change the tree style by using this option S. You can have cladogram, which is V shape. You can have phenogram, branches and squares, curvogram, urogram, swoopogram, circular tree, whatever you wish. Okay, so let us keep it by default, that is P, that is phenogram. Okay, are we supposed to use the branch length? Well, let us keep this branch length. That would indicate which of the sequences evolved first and which of the sequences evolved later. Okay, so everything else is needed to be kept by default, whatever they are. Okay, let us hit enter, and yes, now it says that the plot is written in the plot file. It has done, and we can press enter to quit the program. Okay, so since this plot file do not have any extension that is recognized by Windows based system, we first need to give some extension. I would rename it and give an extension of PNG. You can give JPG or TIFF, whatever image format you are comfortable with. Okay, so now it is recognized by my system. Let us preview it now. Yes, here we have the final phylogenetic tree. Okay, so this is the same tree as we have seen in our consensus. Right, we have this HC1AH, HC1A1, and but here we need to provide some reliability weights as I have shown in this figure. Okay, we need to provide this reliability weight, and for the sake of simplicity we would also tell from what country the strain has originated, right? So what we can do is that we can now open this drawgram, edit this plot file. It would open this in your bitmap. Okay, and all we need to do is to check out in our tree, and we have got the weight of 97. Okay, I have copied it, right? I will use this text option over here, paste it. Okay, you can change the font. Okay, whatever you wish. You can change the size appropriately. Okay, you can make it bold, unbold it, right? So this is it. So over here we have got 97 times this HC1AH has been found to be 97 times close to HC1A1, and similarly we have got the reliability value of 100, which would indicate that 100 out of 100 times this HC1BJ1 has been found to be associated with these sisters HC1AH and HC1A1. Now let us check from where these sequences have originated, right? HC1AH, let us find in our HC1 HC1A1. Where is this HC1AH? HC1AH. A. A is from USA. Okay. So we need to again edit. Let us place USA over here times New Roman. Let us change it to 16, bold. That's it, right? So this sequence is from USA. HC1A1, we know the very first one, HC1A1. It is also from USA, right? A stand for USA. We would again put it over here. HC1BJ1. HC1 BJ1 is D, right? And this D is from Japan. All right. Here we have this one is Japan. In this way, you can kind of specify from where this strain is from, and finally you end up with something like this. Okay, now how do we analyze this tree, right? As you can see, you can save this tree, okay, and you can visualize this tree And you can clearly see that this particular sequences has been evolved in three clades. This would be clade 1, this would be clade 2, and this would be clade 3, and the results are quite similar that are present in this paper. Okay, so this would be clade 1, this would be clade 2, this would be clade 3. Okay, so now how do you interpret this tree? Okay, so this tree you can interpret that this tree you can interpret that the strain from Japan, Moldova and Italy, they have got a common ancestor. Okay, so all of the hepatitis C virus 2B strains, okay, and 2A strains, they are originating from Japan. Okay, while 2K and again 2C, one of the 2C strain is from Italy, and the remaining 2A, or say for example this 2C, 2A and 2B strains, most of them have common origin, right? But the problem is that 2B and 2A are originating from Japan, while 2K and 2C are present in Europe. Italy and Moldova are the countries in Europe, right? So now you need to figure out as to how these things might have happened. So if you trace back to the history, well, this it was reported that there was bidirectional transfer of labor from Europe to Japan, and it is entirely possible that in that event the strain can be transferred from Europe to Japan, and hence these two strains evolved such a way. Okay, and this is also supported by higher bootstrap values. So what you need to discuss in such paper is that how things might have happened, or how this population might have shifted from one position to another position, and how they might have evolved. You can read this paper in more details and find out more details as to how these viruses, Japan is on another side of globe, while Europe is on entirely different side of the globe, right? But still you have to draw some connection as to how these strains might have evolved, or how these strains might have traveled. Okay, so as you can see, most of the HCBK strain, BH strain, HC BG, BD, they are present in the Asian countries, Vietnam, China and Thailand, right? So this data is perfect. The results are quite consistent. Okay, so our Indian strain is of course most closely related to Indonesian strain, right? So we can say that Indian strain and Indonesian strain might have got a common ancestor. This is how you're supposed to interpret the phylogenetic tree. You can read out this paper and see how do you discuss the phylogenetic analysis in your report. Okay, so this is all I have for you, and this is how you guys generate a phylogenetic tree, interpret a phylogenetic tree. Okay, and that was all, folks. Okay. Thank you for your kind attention. Now I am stopping this recording.